what are some issues with DNA sequencing given this fact? So assume up here at the top where it says reference genome sequence, pretend that that's the actual DNA sequence representation of a chromosome. So that's double-stranded DNA, A's, T's, G's, and C's. The way that genomes are sequenced these days is you break that chromosome up into lots of tiny little fragments, like this one I'm circling down here. So that's a representation of one piece of one chromosome. And then what you wind up doing is you only sequence the very ends, the parts that are shown that are the red boxes here. You only sequence the ends, 35 base pairs or so, of each of those pieces of your chromosome. So it's even worse than what I said on the exit exercise from last class. I think I said you get 100 base pair DNA pieces or 1,000 base pair DNA sequences in a row or something like that. These days, for better or for worse, we get tiny little pieces of DNA sequence, 35 base pairs at a time, at random locations across the chromosome. So in actuality, it looks something like this cartoon. You get this huge collection of random pieces of chromosome, and you only get little tiny sequences, the red parts. How the heck do you turn that into the DNA sequence of a chromosome? What tool do we use? We get all these pieces of DNA sequence. Do you think I sit in my office with a piece of paper and figure out how all of those little tiny pieces of sequence fit together to make a human chromosome 5 that's, what, 150 million base pairs long? You feed it all into a computer and you say, computer, you do your job of figuring out how all of those little tiny 35 base pair long pieces of DNA fit together. So in this case, this is what bioinformaticians, people that do, it's kind of like bioinformagic actually, bioinformagicians, if you know how to use a computer. This is what they do. So they write these computer programs that take those individual sequences and they figure out the most optimal algorithms, right, methods for figuring out how all of those little tiny fragments of DNA line up together. This is called a multiple sequence alignment. As you can see, it's multiple sequences, they're aligned. And at the bottom here, which I'm going to circle in red, this program is reporting to us the consensus sequence. So when it looks at all of the different pieces of tiny little fragments of DNA sequence that were generated in the machine, the DNA sequencer machine, it's telling us this consensus sequence is what all of those independent sequences agree is the nucleotide that should be at any one location. Right? So all of these overlapping sequences have 3 T's there in a row. So that's what's reported as the consensus sequence or the reference sequence or the actual sequence of this chromosome.